Welcome to STEM Paths. I'm Dr. Thornton Larson here at Sul Ross State University, where students unpack papers, different study paths, and the people behind them. Our student host of the day is Michael over here, and Michael will be talking with our guest, Dr. Jason Strickland, on his paper, Thamnophis sirtalis and their toxic relationship testing for intraspecific venom variation in common garter snakes. Very cool. So, Michael, take it over.
All right, well, thank you so much, Dr. Thornton. So, like you said, I'm here with Dr. Strickland here today. I am an animal science major, and I'll be talking a little bit about this article. So, I initially read it, and it's mainly about how Dr. Strickland investigated in several species of garter snakes and seeing what type of venom and their variation type is affected. And during the article, I did notice that a lot of the times they did have multiple venom types, like if I remember correctly, 3-finger venoms and some more complex ones, like I think it was SRX1, 2. And this study covers how different venoms are affected in different genes and how they are expressed and not expressed. Does that seem like it covers it, Dr. Strickland?
Yeah, that hits the highlights.
No better the highlights than the lowlights, right?
Exactly. Yeah, so the one thing that one of the main questions we did was if there was differences between male and female venom, because in garter snakes and Thamnophis generally, females tend to be much larger than males. So
I did see that, and that was really interesting to me. It was really fascinating how the females had more venom than the males did.
Mm-hmm.
So I wanted to ask, my first question was, what was the initial idea that really got this study into production?
When I did my PhD and postdoc, we started looking, we moved away from rattlesnakes and elapids and started looking at some of the rear fang snakes to find out what sort of venom properties that they had. Thamnophis was one of the ones that we just started to look at towards the end of my postdoc. And I thought during that time, I was like, oh, this would be a really cool genus to study with students in the future, because there's about 30 species. There's lots of variability in their ecology. And there's some really cool literature known about garter snakes and their relationships with their ability to be able to digest toxic newts in the northwestern U.S. So there's some a lot of cool ecological and evolutionary biology questions with this genus, and they're also common in Mobile, Alabama, where I got my faculty position. So sort of a lot of things came together to where a master student and an undergraduate joined, and we thought that would be a really cool project to work on.
That's awesome. So you mentioned how they had a lot of venom variation and the ecological role on how the species and understanding their sources of introspective variation. How does that adaptive trait, like the venom, play an important role in your research?
Venom is a tool for snakes to be able to communicate with other organisms in their environment. So it's used in both defense and offense for prey capture. And so one of the questions with rear fang snakes, because of their delivery mechanism, they don't have the ability to sort of bite and let go like a rattlesnake does. And so they have to chew on their food items. And so we expect that their venom is used much less for avoiding predation, so basically to scare off other predators. And we think it's more for prey capture and potentially digestion. And so that was one of the things we were interested in with the rear fang snakes, is trying to be able to isolate the idea of a defensive venom and an offensive venom and try to find out what role that played to see what components that they had in their venom.
I always love that description, too, of the idea of a snake, in order to put venom out for rear fangs, having to chew on something. Just the idea of snakes in general chewing.
Yeah, so the other thing about garter snakes and other rear fang snakes is that their venom glands are not surrounded by muscle like you think of a rattlesnake. So those species can contract the muscle and sort of like a needle inject venom, but the rear fang snakes, garter snakes, hognose snakes, several others, it's much more of a passive secretion that goes down almost in the drool of the snake as they're chewing. and that's how it's introduced through the wound. And so there's a lot more.
Kind of like a passive movement.
Yeah, it's much more passive, and there's a lot more to learn about how the venom actually functions. And then it may be not even that useful for garter snakes. We don't know, because a garter snake can swallow a frog in seconds. So the time for that venom to be able to activate is small. So we're not sure the biological function of it, but they're producing toxins that are homologous to their rattlesnake and elapid cousins.
So then you mentioned how they didn't have as much control. So it sounds like rattlesnakes have the muscles to control the poison because they like to go more the offensive, right? Especially because of their environment. Whereas garter snakes have their rear fangs, so they don't have as much control. So it's not necessarily the bite itself that does the killing, but more the passive release of the venom into its prey.
Yes, and the difference is how quickly that can happen. So with the rattlesnake, if it bites a potential predator or a prey item, it's going to be able to inject a large quantity of venom very rapidly, like just getting a shot from a needle. Whereas in the garter snakes, they have to essentially chew on the food item, open wounds, and then that venom is going to seep into the wound of the prey item.
So something I'm a little curious about is you mentioned you'd worked with, elapids and, crotalids and stuff in the past. What are some of the challenges in working with a venom collection in rear fang snakes versus front fang snakes?
I would rather collect venom from a rattlesnake any day of the week. With the garter snakes, you have to anesthetize them using isoflurane, which is a gas that sort of lowers their respiration and causes their muscles to release so that they're kind of limp. And then you inject them with a chemical called pilocarpine. Pilocarpine is used in some eye drops and eye medicines to maintain moisture levels in the eyes because it causes mucus secreting glands to sort of release liquid. And so what the pilocarpine does is it causes the Duvernay's gland, which is the gland that produces venom in the garter snakes, to start secreting. And that's why I said earlier that they sort of start drooling. And we attach capillary tubes to the back of their teeth. And so as that is going down the groove of the tooth, it goes into the capillary tube. And so the challenge with most garter snakes is that it takes at least 30 minutes to collect venom from them. And it's not anywhere near the volume that you get when you collect venom from a rattlesnake and things like that. There are some rear fang snakes that produce copious amounts of venom. The one in West Texas is the lyre snake that you can get a lot of venom from in a short amount of time using this process. But garter snakes in general take at minimum 30 minutes to process, where a rattlesnake or coral snake can be milked in 2 or 3 minutes once you get them secured.
So it kind of takes a little more work with the rear fang snakes.
Yes, but the chance of a student getting severely injured is much lower with the garter snakes. So the reason I don't do as much work on rattlesnakes anymore is because the students in my lab, and the lead author of this paper was an undergraduate student. It's much more challenging for me to be able to work with front-fanged, medically relevant species than it is to be able to work with rear-fanged snakes. So the safety and everything is much better, and the students get to ask the same questions. They get to learn the same techniques. It just happens to be on a garter snake as opposed to a rattlesnake or something else.
And how did you approach the student leading this project in this sense? Because you've mentioned this a couple times, and you talked to me about it when we were talking through correspondence about this was a student-led initiative project, and you're really thrilled about that aspect of it. So I'd like to know a little bit about getting that off the ground, making sure the students invested properly into the idea of the project, all that jazz.
So the student, Grace Commander, is the first author. She's currently a PhD student at the University of St. Louis in Missouri. And so she was in my class her first semester, and she asked if I had any space for undergraduate researchers. This was early on in my position. I'd not been hired for very long, and so I was still establishing my research program. and another faculty member on campus had been doing reptile and amphibian surveys for many years. And the most common snake on campus is garter snakes. And so we started talking about that. So she started helping with the reptile and amphibian surveys, started catching garter snakes, and asked if she could work with them for her honors thesis. So she was able to catch a bunch of snakes on campus, which was nice because we were able to check the traps every morning and afternoon here on the University of South Alabama's campus. So then she collected the snakes that she needed. She got her male and female sample size because her main question was, do males and females have different venoms given their sexual dimorphism in size and behavior? And she took ownership of it. She did the venom collection, the field work, the venom collection, the RNA isolation from the glands, the library preparation for next generation sequencing, and analyzed the data. So she was with me for 3.5 years and was able to be involved in each part of the project.
That sounds like a fantastic student. It always feels great when the student takes ownership of their project. It's sometimes can be a challenge with where interest meets, actual
determination.
Outcomes. Determination. I like that.
And because she wanted to go to graduate school, she knew that she needed this research experience. She started with me early, which was really nice. I always advocate for undergraduate students to get involved as early as possible. We can teach you everything. Even if you're a freshman, you haven't had any knowledge in classes, we can still teach you that information. The only thing we can't teach is that determination and motivation.
Yeah, which I could see could be hard for some college students to do.
Yeah, it's very dependent. We have a small department. We only have 7 research faculty and 400 majors. So there's no way that every student can get involved in research. But the students that are active and interested, you should always ask and try to participate.
Absolutely.
And also from what I've read when I was reading the article, I noticed that she did a really good job about the details. I mean, especially for me, because I've noticed that throughout my life, I'm not a active reader. I don't like it very much, but that article really did hook me and kept me entertained, kept me busy, kept me asking those questions, and it was really fun to read it.
No, she did a really good job. And just like any paper that ultimately gets published, it goes through lots of co-author reviews, collaborator reviews. It went through, obviously, peer review for the journal. So she got lots of feedback. Overall, it was a pretty smooth process. And we didn't, so I guess the surprising part was there was not as much variability in males and females as we expected. And it was a little bit harder to tease apart the venom variation that was due to sex-based differences versus size-based differences. And so because males have a maximum size of 1.5 feet, you don't have any samples that you can compare with the large females to look at their differences. So the male size range limited our ability to really analyze the data as much as we hoped.
Yeah, I noticed that in the charts that there wasn't that much variation in that.
Yep, the males reach sexual maturity and then basically stop growing.
Yeah. So did that make it harder with their smaller size, did that make it harder to catch the males than the females, since the females be bigger?
It didn't make it harder to catch them in the grand scheme of things. We just had to do it differently. So we had drift fence arrays on campus that had funnel traps set next to them. We caught a lot more males in those funnel traps because the males tend to be moving around a little bit more, likely in search of females. Yeah. Where our success in finding the large females was much better just walking around in the environment and
flipping
sort of on the edges of habitat and being able to find them that way. So early on in the project, Grace was catching a lot more males than females, and we weren't catching, like I said, any of the full-grown large females. But when we started doing our habitat sampling and just walking around, we were able to increase our large female sample size.
That's pretty cool. And so did you start early in the morning and then go all the way until nighttime, or did you take breaks in between trying to find them?
The garter snakes are generally active as it warms up early morning. So we were able to between 9 and 11 was a really good time to find them. And then occasionally we would go out later in the evenings, but for the most part, it was like mid-morning was a good time to when whatever makes snakes decide that it's a good time to move, whether it's a combination of temperature, humidity, barometric pressure, whatever that magic combination is, happens between 9 and 11 in Mobile. So
Great. No super early mornings.
Yeah.
no, or really late nights. 'Cause
No, yeah, the really late nights.
like where y'all are at in the desert,
Yep.
you want to go find snakes in the summer, you've got to start at like 10 p.m.
Yeah.
and like look around in the evening because there's so much, it's so hot there that they wait until much later to move around.
Yep. And then, yeah, that's what, summertime, you see the cars on the side of the road flashing the cuts, looking for that Alterna.
Exactly. And I teach a field course out to West Texas every May semester with a botanist. And so we were in the month of May this year, those people on the side of the road with our safety vests on, looking at critters.
Oh, I should talk to you more at some point about that, because I know the Texas state system were finalizing a new research station over in the Christmas Mountains.
Okay.
And they're going to be looking for people to want to bunk there for, while they're doing research here.
Fantastic. Yeah, we stay at Black Gap Wildlife Management Area.
Oh, Black Gap's great.
And then we stay at the Nature Conservancy property in the Davis Mountains.
Mm-hmm.
And then I think Midwestern has they have another field site out there somewhere near Alpine as well.
Yeah, everybody likes to come out here.
So we got all the
But
wildlife.
I'll happily take you up on any opportunity to come bunk in the desert and look for critters.
Oh yeah, no. Let me know when you're in town. I'm happy to hang out and look for snakes. When do I say no to that? What was the biggest frustration of this project? When did it hit a point where it was like, oh God?
Oh, that's a good one.
Or was this one just the ultimate beautiful project where there was never a dang it moment?
No, the frustrating moment was when we got to the end, we had the data analyzed, and we were starting to generate the graphs and doing the statistical analyses.
Yeah.
And pretty much the first time we did it, everything we came back as not significantly different.
Mm-hmm.
So there was like no positive results. And that was frustrating because we expected a pretty clear difference between males and females. Because like I said, the females are 3 times the size of the males.
Mm-hmm.
They potentially eat larger food items. They have mammals in their diet occasionally. So there's lots of reasons to expect that the venom is going to be different. And so we did some simple t-tests, and we weren't able to find those differences because we just had male versus female. And it wasn't until we started accounting for the ontogenetic change, which is known in venomous snakes, that ontogeny, which is change over time as they grow and mature. We know that in rattlesnakes, there's ontogenic variation. We know that there's ontogenic variation in other venomous species. And so we had to, what we thought was going to be a really simple, straightforward analysis, we had to code in a few more factors to be able to account for the size-based differences as well. And once you accounted for the size, you got to see some sex-based differences. It still wasn't to the extent that I thought was going to be there. I thought it was going to be a really straightforward result. And we still never got, here's this p-value of 0.0005.
Oh no, that one.
Flashing differences, nothing like that. It was much more subtle than I expected.
Right. And on that, maybe you can think on talking about a little bit of that bias we see in publishing, where you have to have that result to publish versus the no difference being just as important to a certain degree, as long as you've got your variables in check.
Absolutely. So this paper was going to be publishable regardless. One, it was the first time that anybody had really looked at Thamnophis venom glands for the genus. So just as a descriptive study saying, here's the venom components of this species, would have been a publishable result. And if we would have found no sex-based differences, that would have been completely okay, and I think it would have been all right. But I know you're not supposed to, but you have sort of expectations are going to happen.
Well,
And
that's that educated doing your background search to understand that you observe these differences, so you're trying to seek that in a more clear way.
Exactly. And so obviously the stats bore out that the result wasn't nearly as distinct as what we expected. But we learned some cool things about their biology. We started to question some of our assumptions initially, and I mentioned it earlier, we don't know the true biological function of this venom.
Mm-hmm.
We can inject it into prey and show it does certain things. We can run assays on it and know that it has these certain activities. But in reality, when a garter snake, I watched a garter snake eat a frog, and the thing was down in 5 seconds. It's there's no way that there's enough time to
let that
chew on it, introduce the venom, and have it have some sort of biological effect. Unless potentially, once those toxins get introduced, they may help with some sort of processing of the prey during the digestive process. But that's almost impossible to actually test. So
it almost could be that the venom is almost like a backup plan sometimes for the garter snakes.
Potentially. And so I know hognose snakes that specialize in eating toads have really large back teeth. They'll use those to puncture the toads as they air up. And I would assume because the toads fill up with air, I've seen a hognose take a lot longer to be able to swallow a toad. And so in that case, maybe the venom does cause paralysis in the prey item. There's enough time to introduce it, both because the toad is struggling and harder to hold down. And so maybe that does actually help the hognose swallow the toad. But for the garter snakes, most of their prey items are not that challenging to manage. And so it brings up some cool questions about prey handling and for different species, but not something I've been able to look into.
That's really cool. I mean, it's interesting how some snakes would catch their prey, especially with the garter snakes, relating to how they use their venom and stuff. It would be interesting to see if that venom is a backup plan or is just there to secrete, like you said, to help digestion.
Well, and if you think of most snakes, snakes don't tend to have much defensive mechanism. They don't have arms and claws or any of that kind of stuff. And so one of the reasons we think that venom exists is because it limits the chance of retaliation from the prey item. So if you are a rattlesnake and you bite a large squirrel, if you hang on to that squirrel's going to bite you back, and it's going to cause major injury. But if you can bite it, let go, and then let the venom do its job, then great. But the prey that the garter snakes eat don't have the retaliatory capacity, as far as I know, at least the ones that we've noticed them eating here on campus.
So I'm going to pivot just a smidge here toward more of a career-focused thought of, so did you always see yourself getting into academic research, or when did you make that a goal and accomplish all that, and how did that all play out for you?
So the short answer is yes. I've always wanted to work with reptiles particularly. And as I'm a first generation college student, so I got to undergraduate at Angelo State University in San Angelo, so not far from Sul Ross.
I know it well.
And so I did undergraduate research there and realized that, okay, you can sort of get paid to go catch animals if you're an academic. And so I started working through there. I do regret that I never considered sort of other options as I was going along. I had very much blinders on the entire time. I did my undergraduate, did my master's, PhD, postdoc, faculty. Now that I'm a faculty member trying to mentor students on fields outside of academia, my knowledge base is not as good as I would like it to be. So I'm having to learn a lot more about what's required to be in a non-governmental organization, what's required to be a state biologist, what's required to do these different agencies, or going into consulting and things. So I'm having to learn a lot more now because I never considered any of those paths as I was progressing through my academic career.
Oh, wow. So during your research fieldwork and your data analysis, you mentioned some of the challenges that you faced while researching it. What were some of your favorite parts about it?
I mean, aside
So
from
I think-
catching the snakes, of
Yeah.
course, you know.
Right. I was thinking it's like it's anytime you sort of reach a goal or you see the student having some sort of excitement. So like we had set a goal for catching 10 males and 10 females, and we got that goal. And then, like, so the last snake for the undergraduate student, like she came in, Grace came in super excited, like we're done, like we've got our field work finished. And then when we got the sequencing in, and like she hit the command to look at some sort of sequencing file on your computer, but she used CAT, which is basically just prints out your data on the screen. And so she was able to see all the hard work on as A, T, C's, and G's as they came up on our computer screen. It's like, okay, I caught that snake, I collected its venom, I processed it, I got the glands, I did all the lab work, and now here's the data. Like I can see this is the, in this case, the mRNA sequence for this particular species. And so it was really exciting. It's I get more excitement about student success than necessarily my particular thing.
Well,
And then
I think
obviously when the paper got accepted, that's super exciting.
Oh, absolutely.
Yeah.
No, I think when you get student success, it starts to really be like, oh man, I can do this a little bit. I can help students reach their own goals and find them in different ways. And watching them, like a recent experience I have with a grad student of mine, he's finally doing his field work after a long time of working through his proposal and all that stuff. And all of a sudden you see a hop in his step again, because he's finally, all the work he put into the experimental design is finally starting to bear a little bit of fruit. And I expect it to keep going as he continues to progress. And that is a great feeling, because some of those days when you're doing the experimental design can be really, can be wear you down quite a bit.
Yeah, so it's like feeling that feeling of like, hey, I actually did this. Like I can actually complete this project.
And seeing someone else do that too is very positive.
Yeah. Yeah, I don't have research myself, but I do remember where I've had to do some major projects back in high school and for some college classes, and it's that feeling of like saying, I did that, like I can do this process, and just seeing that spark of fire to learn more about science and learn more about whatever it is you're researching is a really good feeling.
Absolutely.
So I think we're at a point where we can probably start thinking about wrapping up. So do you want to run them through the last couple questions we have? These are a little more broad-based somewhat.
Sure, yeah.
But
So what's one piece of advice you'd give your undergrad self?
you kind of touched on it a little bit, but I think there might be more.
Yeah. Advice I'd give my undergraduate self. That's a challenge. Yeah. So I think one would be to, as I mentioned earlier, to think about other career paths and not be so focused on what I was trying to do. The other thing is I wish I would have focused a little bit more on, I have a problem with, and I guess I still have that problem as a faculty member, of doing too many things at once.
It's hard to say
so
no.
It is hard to say no.
Yeah.
And so I wish as an undergraduate, I would have developed those habits to give my all to a smaller number of things to make sure that they get finished, instead of now dragging on several things that are incomplete that I started but didn't finish. And so I set those habits the wrong direction, and now they've carried all the way to me now as a faculty member.
Yeah. And so for everybody out there that's doing research in science and all the scientists out there, what's one thing you wish more people understood about this?
About science and scientists.
Yeah.
The amount of passion that we have for the things that we're working on, and that there are no ulterior motives for why we're doing what we're doing. It's that we want to increase knowledge in the world. We want to be able to do the things that we enjoy, that contribute to basic science. And I know there are many researchers out there that work on application-based research, but even the people that are working on garter snake venom or other things that are just trying to describe the world around us have lots of potential great applications. And so the classic one recently that's gotten all the attention in the venom world was the the early work on Gila monster venom has now led to what we see in the weight loss drugs and diabetes treatments and like Wegovy and Ozempic and stuff like that. So it was basic science 20 plus years ago that led to these discoveries that are now helping a lot of people. And so those researchers had no ulterior motives. They were trying to just understand and describe the world around us to increase our knowledge. And so
when
scientists are...
Yeah.
Go ahead.
When I'm explaining to my students the difference between applied and basic science, and then I talk about how the bridge from basic science leads into serendipitous discovery, you know?
Absolutely. And with the garter snakes, we have, up until this paper, we had very little knowledge about their venom proteins. We know in rattlesnakes that the venom has been used in over a dozen treatments for different diseases and disorders. We have no idea what's in garter snakes or tarantula venom or scorpion venom or a lot of these other species. And not that we should be using them for sort of this bioprospecting idea, but who knows what we can learn from the components and the biology of these different species that exist.
Well, understanding these protein cocktails helps us to think about how to synthesize them in a lab in a way that
Absolutely.
they can work. I'm not thinking of, we have snake farms for medical science, which we do for some of that stuff, but from a get this basic stuff running, and then you get into the synthesis of proteins in order to create a different process of developing medical treatments from those initial proteins.
Yeah, and
Right, absolutely.
it also amazes me how we get to also see into the little things that go into possibly making the venom. So the DNA, the RNA, all of that process of making the actual protein that makes the venom for either a snake or tarantula, like you said, scorpions. It's really interesting to see how that plays a role.
So go ahead. Sorry.
Absolutely. And so that's where my research has sort of moved into evolutionary genomics, is really understanding the process of transcription and translation. Because if we can understand that process, it helps us, that's a foundational biological function of every cell in our body. So the more we can learn about that, the better.
Absolutely.
Yeah, exactly.
And the very last question, this might be hard for you based on your what you've said about your career ambitions so far, but if you weren't doing this, what would you be doing?
That's actually a very easy answer.
Oh, he's got it.
So I would probably own a restaurant. So my only hobby is taking my Texas roots and bringing them to the southeastern U.S. And so barbecuing brisket, pork, beef ribs, all those kinds of things. And so my main hobby is barbecue. And so we catered a wedding for the first time not too long ago, and it was a lot of fun. And so if I was doing anything else, it would be owning a restaurant that had some sort of barbecue component.
A nice brisket, huh?
Nice brisket restaurant. So would it just be for brisket, or would it be the whole Texas plate?
I would do the whole Texas plate, but bringing it to other places. And then there would be other things besides barbecue, because barbecue is a lot of work and time and things like that. So it would be, so the brisket and the barbecue would be probably like the special on Saturday, Sunday kind of meal. Whereas I don't know what the focus of the rest of the restaurant would be, but it would be a lot of fun.
All right. Fantastic.
That's very cool.
So I want to take this moment to thank Dr. Strickland here, Dr. Jason Strickland out of University of, what was it, Southern Alabama, right? South Alabama?
University of South Alabama.
Right. Just want to make sure. University of South Alabama. And thank Michael for looking up this paper and saying, hey, I want to talk to this guy, and making up all these questions that led us down this really cool conversation. So this has been STEM Paths, where we unpack papers and the people behind them. And I think that's been done really well today. So thank you. It's been such a joy with all of you.
Thank you very much.
And I hope to talk with you both more. I mean, obviously, you're stuck in my class. But Dr. Strickland, I hope to talk with you more in the future, too, especially since you like coming out this way.
Thank you all very much for having me, and I look forward to those conversations.